everyone, welcome back to class. Today we're going to continue our talk on glutamate. We're going to start by talking about the rest of the glutamate receptors we didn't cover last time. And then we're going to talk about the systems that glutamate is involved in. So, our metabotropic glutamate receptors. Now remember that these receptors are going to be slower than the ionotropic receptors. However, because they're glutamate, they're still going to be faster than some of the other neurotransmitter receptors. Okay. Now I mentioned before that there are three different groups of receptors here, group 1, 2, and 3. Group 1, this is mGluR1 and mGluR5. Okay, so M for metabotropic, glutamate receptor 1 and 5. These are GQ receptors, and so they lead to activation and the roles of PLC and calcium. Group 2 receptors. These are mGluR2 and 3. These are GI receptors that decrease CAMP. And then group 3 is mGluR4, 6, 7, and 8. And these also are GI coupled receptors that decrease CAMP. So group 2 and 3 lead to the same thing, but they are two different groups. Okay. Now, group 1 of these receptors are mostly found on that postsynaptic cell. These are GQ, but groups 2 and 3 are often found presynaptically. And you can kind of guess that because they are GI receptors. And a lot of GI receptors act as autoreceptors. And so they help modulate other neurotransmitters within the same neuron. Because remember, a glutamatergic neuron can also release other neurotransmitters. So say a glutamatergic neuron could also be a cholinergic neuron, for example. Okay. These receptors all help contribute to the plasticity of synapses. And these can be excitatory or inhibitory depending on signaling and cell types. But remember that glutamate itself is going to be excitatory, okay? But the receptors can be either excitatory or inhibitory depending on what they do and what cells they're acting on, okay? So whether they're autoreceptors, whether they're presynaptic, postsynaptic, what kind of cell is after them, that kind of thing. There has been a number of knockout studies to see what the role of these receptors are, what would happen if we didn't have certain metabotropic receptors. And so mGluR1, so remember that's one of the group 1 receptors. If we knock out the genes for this receptor, then we see motor dysfunction. So we see ataxia, intention tremors, and dysmetria. And specifically, we see impaired plasticity in the cerebellum, which the cerebellum does play a role in your motor movements as well. So there is a role for these receptors. You do need them. You can't just rely on your ionotropic receptors. mGluR2 knockout, we see normal synaptic transmission. What we see is that this knockout really only reduces presynaptic inhibition so that it plays that role as the autoreceptors. Okay, so transmission is normal, but we get kind of extra release. We don't get that feedback that we need. mGluR4, we see loss of synaptic efficiency during repetitive activation. So that presynaptic regulation in the cerebellum and maintenance of normal motor function. is where we're seeing this loss. Okay, so it has to do with that plasticity. So how do all of our receptors look within, say, a cell? So here is just a diagram suggesting what things might look like. So at the postsynaptic densities, so on the postsynaptic neuron, this is what the receptors might look like, or the distribution of the receptors might look like. What we see is that our metabotropic receptors tend to be on the periphery of that synapse, and our ionotropic receptors are going to be mostly in the middle. Okay, so here you can see in red we have the NMDA receptors, and in white we have our AMPA receptors, because remember, you can't have NMDA without either AMPA or kainate receptors. So they're pretty well distributed. But what we also see is that a lot of these NMDA receptors are coupled with CAMK2. Okay? And so this is a calcium-dependent protein that we're going to talk about in terms of plasticity. And so the role of the NMDA receptor can be closely tied to the role of CAMK2 in plasticity and learning. But just in general, the density, so say this is where the synapse connects. We're going to have our ionotropic receptors, a lot more of them in the center of that synapse, and then on the periphery of the synapse is going to be our metabotropic receptors, most likely group 1 metabotropic receptors. Okay. So moving on to plasticity. We see plasticity in lots of parts of the brain. It's very important for normal function, but especially important in learning and memory. We see a lot of plasticity in the hippocampus, which of course is the area most associated with learning and memory, but we do see it in other places around the brain as well. Synaptic plasticity, just the definition of that, would be changes in the strength of glutamatergic synapses in response to activity. So long-term potentiation is one option. The other option is long-term depression. Okay, so these are the two types of synaptic plasticity that's possible. So long-term potentiation is an increase of the synaptic strength, and long-term depression is a decrease in the synaptic strength. Okay, so you'll see that long-term potentiation, it says it's following tonic activity, so that's a lot of activity very quickly, a lot of intense activity very quickly. Long-term depression, you can see, is slow, repetitive activity, so low intensity over a long period of time. Okay. Now, it is important to note that LTP and LTD are only described experimentally. We haven't really been able to demonstrate them like in a fundamental process in either animals or humans. We really just describe it, but we kind of agree it's happening. And this is what we use to explain the changes in plasticity and the changes in learning and memory. Okay. So how does this relate to our receptors? Probably wondering. Oh, I'm jumping ahead. Okay, so plasticity in the hippocampus. So here's your hippocampus in a rat. This is what it looks like. Okay. Hippocampal plasticity is widely studied. Because there's so many circuits that we know a lot about. We know the CA1, which has those pyramidal cells. We know CA3. We know the granule cells within the dentate gyrus, etc. So it's very easy to study, especially in rats, this whole system and how everything connects and how we can assess the strength of the signal being sent from one to another. You can do ex vivo preparations. You can do extracellular or intracellular recordings here. It's just one of the areas we use to study plasticity of the hippocampus quite a bit. So, LTP occurs through what we call coincidence detection. So pretty much what happens, so this is an example of a stimulant or of a glutamatergic synapse. So up here we've got that presynaptic glutamatergic neuron. Here are vesicles containing glutamate. On the postsynaptic cell, we have an AMPA receptor and an NMDA receptor. Okay, when only a little bit of glutamate's released, it will act on AMPA receptors and pass through. So sodium's going to pass in. Okay, glutamate will bind to the NMDA receptor, but there's not going to have been enough of a depolarization from just, say, one AMPA receptor or a single pass of the AMPA receptor to depolarize that membrane to allow the magnesium block to be removed from that NMDA receptor. Okay, so a single stimulus, you're only going to get AMPA receptor activation. Now, after a while, or if you have repeated stimulation very quickly, what we call tetanus, then you get repeated quick activation of these AMPA receptors, which causes that depolarization across the membrane, which then allows that magnesium to be removed from the NMDA receptor. And now calcium can enter the postsynaptic cell via that NMDA receptor. And if this happens enough, that can then lead to LTP. Okay. Now, I mentioned CAMK2, which is coupled with NMDA receptors. So calcium calmodulin-dependent protein kinase 2 is the full name. It's a protein kinase. I mean, that's really all it is. But it's localized within those NMDA receptors on the intracellular phase. So it's within that postsynaptic cell, connected to the NMDA receptors. And what happens is it phosphorylates a number of different cellular targets and initiates what we call early phases of LTP. So when you activate the NMDA receptors, it activates CAMK2, which then activates LTP. So here's exactly what's going on. This is early stage LTP. So we've got our AMPA receptor. This is the original AMPA receptor. We get tetanus of this receptor. Lots of repeated activation that leads to the removal of the magnesium in our AMPA receptor. Calcium flows in. That's going to then activate the CAMK2. Okay, so this calcium calmodulin 2. And what it's going to do is it's then going to go over here and it's going to tell the postsynaptic cell, hey, we've got lots of activity going on. Let's increase the number of AMPA receptors in this synapse. And that's what LTP pretty much is we're increasing the number of AMPA receptors so that we're going to be able to more quickly activate the NMDA receptors in the future. Okay, so right now, or originally, you only have, say, one receptor, so it's going to take a lot of repeated activation of the one receptor, and that could take a while to activate NMDA. But if we have, say, 3 or 4 AMPA receptors, then we could get that depolarization that's required quicker, therefore activating NMDA quicker. Okay, so that's what happens in LTP pretty much. I have other things described over here. We'll just go through that quickly. So CAMK2 will phosphorylate the AMPA receptor, increasing their sensitivity to glutamate. So say if before they needed 2 glutamates to bind, they now only need 1, that kind of thing. The signaling will then cascade, increasing the trafficking of AMPA receptors to that postsynaptic density. That increases the availability of receptors. So now we not only have this increased sensitivity, like we have here, but we also have more receptors, okay? So the receptors that were already there are going to depolarize even quicker, and now we have extra AMPA receptors. We are also going to create some retrograde messengers. And what these do is they actually signal to the presynaptic cell, initiating presynaptic changes that will also release more glutamate. Okay, so the NMDA receptors are huge. They tell current AMPA receptors, hey, phosphorylate quicker, depolarize quicker. They say, hey, let's get even more AMPA receptors, and then they send a messenger to that presynaptic cell saying, hey, we need more glutamate. Okay? So all in all, this is LTP early phase. Okay, we're going to get more depolarization of that postsynaptic cell. Late phase LTP, then we're going to get activation of CAMK2 and PLC, which will then converge on another signaling kinase called ERK, or extracellular signal regulated kinase. And this triggers downstream changes, including phosphorylation of transcription factors, which can then lead to the synthesis of even more AMPA receptors. Okay, so early phase is saying, hey, you know those AMPA receptors you have in storage, let's put them on the synapse. Late stage is taking ERK and telling the cell we need to make more AMPA receptors so that those new AMPA receptors can also go. So the synthesis process is very important for maintaining this LTP, because now we have lots of AMPA receptors pre-made in storage to replace the old AMPA receptors that maybe, they have a shelf life. Eventually they need to be replaced. So now we don't have to wait to replace them. We can replace them as soon as possible because we have these brand new AMPA receptors. So that's how we maintain that LTP. So LTP, very important with NMDA receptors. The induction depends on NMDA receptors. Okay, we need those NMDA receptors in order to initiate LTP. What we've seen is that in mice, we can look at overexpression of the receptors actually will increase learning in mice. So rats that have more NMDA receptors will learn things quicker than mice that have just a normal amount of NMDA receptors. Okay, so here we just see it. Mice are will over-engineer to overexpress a certain subunit that leads to NMDA receptors. They're called duty mice. And here is a learning task. So they learned something originally. After 1 hour, how much do they still remember it? And then not remember it. And what we see is that our two Doogie mice, so you can't see, but there's two lines here. They're just overlapped. They are remembering things for longer, whereas the wild-type mouse is able to remember things but kind of loses it more quickly. Okay, so the memory and learning is quicker in these Doogie mice. Okay, let's stop that here, and in the next video we will start talking about excitotoxicity and cell death.